All right, we're just going to start, even though the other competition isn't really over. So Dr. Cuellar attended college at the National University of Colombia, where she became interested in crops biology. She moved to Mexico for graduate studies at UNAM's Biotechnology Institute, where she obtained master's and PhD degrees in 2001 and 2008 in 2008. In grad school, she focused her attention on cell wall proteins, and carbon metabolism. In 2009, she was also accepted in the postdoctoral program at UC Davis, where she continued her studying in plant adaptation to stress and entered the world of genomics and large data set analysis. Please give a warm welcome to Dr. Cuellar.
Thank you, Sophie. I'm gonna share my screen and I'm gonna show you a couple of things. So, as Sophie said, my name is Sonia Cuellar. I work at SCHMAHL Science Workshop, where I am a mentor for teenagers that wants to learn science and do some research in science. Today we're going to talk about shape, function, and proteins and how those things are related. I want to call your attention. I have here in the presentation this white screen. If you have the opportunity, you could use that space to pin the screen in which I'm being shown and increase the size of that screen so you can see me a little bit bigger than in the small squares, more screens of the divided screen. That is because I'll be showing you the presentation, but I also want to see, I'll be doing some things with my hands and I want you to be able to see them. So thanks again for inviting me. And I'm gonna start by setting an experiment. We are gonna talk about the experiment over the time of this talk, but I want to begin by setting the experiment because it will take a little bit of time for the things to happen. So I want to be sure that at the end, we can observe the results. So for doing that, I have here 2 cups that I hope you can see. I labeled them. This is cow milk and this is almond milk. I have half a cup of each one here in my cups and it's warm. It should be warm enough that, let me remove this. It should be warm enough that when I touch it, I feel it warm, but it's not hot. I'm not burned. I can touch it. And what I'm gonna do is add to each one of those cups, I'm gonna add half a tablet of rennet. This is a substance that is used for making cheese. It used to be extracted from the guts of cows, but now is prepared using transformed bacteria. So we don't need to feel bad about the little cows when we use rennet because they are not involved in this anymore. We use bacteria for that. So I have here half a pill that I already crushed to powder, and I'm gonna add it to the cow milk. And I'm gonna crush the other half of the pill to add it to the almond milk. Let me see. Have it here, so I'm just gonna crush it. And the only reason I'm doing this is to make it easy to mix with the liquid. I'm gonna use those squares to mix very well. And I will leave it aside to do its work while we talk a little bit about this subject. We will go back to this, but it takes some time. So we will keep talking for a minute. and then we'll back to observe what had happened there. Now, I want to talk about what we just did. What we use is this Rennet, I was mentioning it, is a commercial product, and is actually a protein. What is packed there in those tablets is actually a protein that it used to be called a rennet. And this is the three-dimensional representation of that protein. Proteins, as you know, or most of you, I think, may know, are the product of putting together, aligning together many different amino acids. So you have a sequence of amino acids that I have represented here. In this case, are the first 30-something amino acids to the rennet protein. The protein actually has 281 amino acids, so they didn't fit here, but this is just the first, and this will be the sequence of the different amino acids. So methionine, oh, I lost my cursor, methionine, aspartic, guanine, cytosine, leucine, valine, valine, leucine, leucine, aniline, valine, phenylalanine, so on. That's the same that I have, a representation of that. It will be what I have here in this pipe cleaner with beads. And so, as you can see in the pipe cleaner and also in the sequence of the protein, we have different amino acids repeat in different order. And that order is what we call the primary sequence of the protein. And it's very important because it is dictated from the genetic information. It comes from the information that is stored in DNA. And it determines the function and the structure of the protein. And the way that works is those amino acids have different properties. One very important of the different properties that they have is they may be what we call hydrophilic or hydrophobic. Some of you may know hydrophilic means it likes water. Hydrophobic means it doesn't like water. So here in the sequence of the rennet protein, I have highlighted the ones that are hydrophobic in purple, and in white, the ones are more hydrophilic. For my pipe cleaner model here, I will say that the orange beads and the pink beads are hydrophobic. And the reason why that's important is because all proteins are in the context of a cell that is an aqueous environment. There is a lot of water around there. So the hydrophobic residues don't want to be close water. They rather be close together. So they are gonna try to be close together. So in my model protein, pink will try to be with other pink or with orange. And sometimes the tension of the protein don't allow them to be that close together, but they still will try. And that property of being hydrophobic and the other hydrophilic, in combination with other properties of the amino acids, but that very importantly will determine the three-dimensional shape of the protein. And that three-dimensional shape of the protein that is given by the interaction between hydrophilic and hydrophobic amino acids is very important because the function of the protein depends on that three-dimensional shape. I'm gonna stop sharing this screen because I'm gonna start sharing a different screen here in which I'm gonna show you the three-dimensional structure of the same protein we're talking about. We call it a chymosin. That's a more technical name. Rennet was the name that was used at the beginning of this 20th century when it was described, but this is chymosin. This is the enzyme I'm talking about, and this is the three-dimensional representation of that protein here. This is a representation that we like a lot because it's showing different parts of the protein, parts that we call lamins and ribbons and helix and coils, and all of that have a meaning. I'm not going to go into those details. I want to show you, for example, that here I have highlighted in white. I hope you can see here this area that is white. That is what we call the active site of the enzyme. This protein is actually not just a protein, it's an enzyme, which means that it has a chemical function. It does something, it transforms something. And it does it in this active site that I have highlighted here in white. As you see, is kind of in the internal part of the enzyme, but it has this groove the substrate can go into and reach that area. All the protein is important for the function, but the actual chemical reaction happens here. This is, by the way, I'm using here the software that it was developed by the University of California, San Francisco. It's a free software, it's called Chimera. And I'm using a database that is called Protein Database, PDB, and it's a free database. You can access there all the proteins that had been, their three-dimensional structure has been elucidated. The three-dimensional structure is there. I want to show you other things that we can do when working with this visualization of the three-dimensional structure. This is a different visualization. It's not now it's showing the different atoms, not the ribbons and arrows, but it's showing the atoms. And you can see that is enzyme there, and it has this kind of crescent moon shape. This is the groove that the substrate needs to get into. I hope you can see there in white inside, in the middle of the protein, is the active site. And I can use this to also see the, oh, sorry, I should have, where is it? I need to select this one because I can ask the software to show me now the surface of the protein, not the atoms, but how the surface of the protein would look. And this will be the surface of the protein. Those atoms that you see there kind of outside of the surface of the proteins are actually molecules of water that are necessary for the protein to configure in the proper shape. And I could also use this to see in the surface where are located the amino acids that are more hydrophobic and more hydrophilic. In this representation, in blue, the more hydrophilic residues, the more hydrophilic amino acids, and in red, the more hydrophobic. So as you see the surface of the protein, this all will be, the black here will be all water, it's inside of a cell or an aqueous environment at least. And so that means the surface of the protein exposes a lot of hydrophobic residues. There is some hydrophilic there that are kept in place by the structure of the protein, but most of the surface of the protein that is interacting with water is actually hydrophilic. And that is very important because that will keep the shape of the protein, will keep the groove necessary for the substrate to get in, and the active site that is here inside. Now, why is that related to all those things that I've been telling you about milk? Why I'm talking about milk? Well, I'm talking about milk because the chymosin is this enzyme that I've been showing you. And what this enzyme does is an enzyme that cuts casein. As you may remember, proteins are in many different kinds. Proteins are the ones that form our hair or nails or muscles and skin. All of that is proteins, but there is also a specific and very important group of proteins that are called enzymes, and are proteins that have a chemical function. They do something. In this case of the chymosin, what chymosin does is cuts casein. And casein is important in this subject because casein is the most abundant protein in milk. I don't have my cardboard of milk here because it's very warm today. I don't want it to spoil, but I put a little milk in this bottle just to be able to show it to you. I know you all know milk, but just to remind you, milk is this white liquid that is very nutritious, and is nutritious because it's rich in protein, but it's also rich in other ions that we need, like calcium and phosphate. And the thing with this milk is very, very stable. I can shake it and it will still be milk. I can centrifuge it and it will still be milk. We can boil it and it will be milk. We can freeze it and if we let it go back to room temperature, it will be milk. We can actually dehydrate it, remove all the water, and it will become milk powder. And when you rehydrate it, add water back, that milk powder will form this white liquid that is it has all the casein on it. Is well, all the other components of milk too, but importantly the casein, that is what it gives the white color to the liquid. Milk is a very, very stable substance, and that's very important from the biological point of view because we mammals use milk to feed our babies, or human babies, or cow babies, or any other mammal. Offspring is fed in this white liquid, and it needs to be very stable so we can carry it with us and have it available for feeding babies. So all that is very good, and I think I'm not saying nothing that you already don't know. You know milk, you know that it's stable. You may not know that we can dehydrate it and form powder and then rehydrate it later, but you know milk. Milk is a very stable substance. The thing with that is casein is actually a very hydrophobic protein. When we have the sequence and we analyze the sequence of this protein, is actually very hydrophobic. You may remember from the slide I showed you before, oh no, that was the chymosin. But casein is actually very, very rich in hydrophobic residues, a lot more than many other proteins that we normally find in liquid form. So that creates question. How is possible that this very hydrophobic protein is actually in water all the time and is very stable there in water? My jar is dripping. So how that happened? And that's a question that we have for a long time, and just in recent decades start to clearly answer. Actually, we don't have yet a three-dimensional structure for a casein. In general, crystallizing hydrophobic proteins is not easy, and you require a crystal of the protein to be able to use X-ray diffraction and elucidate the three-dimensional structure. But we know a lot of things. Even though we don't have the three-dimensional structure of casein, we know a lot of things. We know, for example, and that is an explanation, that casein in milk is actually in these micelles. This is an actually microscopy picture. The diameter of this micelle is around 120 micrometers, and these structures are what is soluble in water. Those micelles are represented here in the this diagram, and they are composed like this. This is the micelle, in which, as you see, is a very complex structure that has molecules of casein of two different types. The K-rich casein that is presented here, that is actually a little bit more soluble because it's in contact with some ions, and that is surrounding the other casein molecules. And all of them are kept together by interaction with calcium phosphate. So this partially explains why milk is so nutritious. It's carrying calcium, it's carrying phosphate, it's carrying the casein. And this structure explains how it's possible that this very hydrophobic molecule is actually very stable in water, because those micelles themselves, they are just exposing the hydrophilic part. So the micelle itself is hydrophilic. The components of the micelle are not necessarily as hydrophilic. And that is very important in relation to our experiment. I want to show you here what happened to our experiment. Let me see, how much time do we have? Yes. So this is the cow milk, and this is the almond milk. As you can see, almond milk is pretty much the same that it was at the beginning. The cow milk, I hope you can see, is not running. It's not running because it has changed. I'm gonna disturb this a little bit to show you. What is happening is the protein has coagulated, or is in the process of coagulating, and is separating from the liquid. I actually have here another cup in which I did exactly the same that I did with this one just an hour ago. And so it's just a process a little bit more advanced. And I wanted to show you this because I hope you can see that the solids are now separated from the liquids. This is the process that we use when we are gonna make cheese. We separate the solids in the milk from the liquids. Let me see, I hope you're seeing it there. And this is because now the Chymosin have disrupted the molecules, the micelles of casein. The micelles of casein are not anymore in their hydrophilic form. So now the casein is separating from the liquid. I hope you can see the liquid there. If not, at least I hope you absolutely can see that this is not a liquid anymore, as the almond milk is still a liquid. This is moving like a liquid. This is not moving like a liquid. This is the one I prepared an hour ago. This is the one I just prepared with you. So what is happening here? Something is changing the structure of the proteins in the milk, and is actually draining the chymosin, because that is what we add. And what the chymosin actually does is it cuts the molecule of casein in two parts, actually. So this is, of course, is a schematic representation. Things don't really look like that. But if you cut the casein molecules, now they don't have the three-dimensional structure that they need, and they start exhibiting more of the hydrophobic residues. That means that they want to come together, and then they coagulate. They are not anymore able to form this structure that will easily and happily interact with water. They form these structures, or many different variations, this is irregular, in which a lot of hydrophobic areas are being exhibited here, and those means that the protein separates from the liquid. What is happening there, if I go back to my model with the pipe cleaner, is remember that the rules for the model is the pink and orange are hydrophobic, so they want to be together. And because of that, they are keeping this three-dimensional structure of the protein. Of course, that is not the only force that is there keeping the three-dimensional structure of the proteins. There is other things related with the other amino acids, but that is a very important component. So if I come here and I cut, and I'm gonna cut here whatever place, but chymosin actually cuts in a very specific place. Now, the two resulting peptides cannot keep this three-dimensional structure. They still have the same hydrophobic residues, and they are attracting each other, but the structure is not stable anymore. That is what chymosin does. And because the structure is not stable anymore, now the protein is exhibiting a lot of hydrophobic residues that before were protected inside the protein. And that means the protein is not able anymore to happily interact with the water and be this liquid there, and start separating from the liquid and forming the solids. If I allow this process to keep going and I use cheesecloth, for example, for filtering this may later become in cheese, and I will separate what is called the whey. It's the liquid of the milk. So here, what we have done is a home experiment in which I hope you can see that there is an enzyme that is working. That was the enzyme that comes in the rennet, the chymosin, and that enzyme is working to change the three-dimensional structure of other protein, the casein, and because it changed the three-dimensional structure, it changed its properties and its function. Its ability to work as milk now is becoming curds that can later be cheese if you want. That is not happening in almond milk. And as you saw, I put the same rennet, both were the same warm temperature, and it's not happening. I want you to stop for a second and think why the rennet will affect the cow milk, but will not affect the almond milk. You may have different hypotheses, and I'm sure all of them are very interesting. And we may be able to develop even homemade experiments to test your hypothesis. I can tell you that is related to the fact that almond milk and cow milk are different. They are different in the source, but they are also different in the components. There is no casein in almond milk. There is other proteins, of course. It's not that there's lacking of proteins. It has proteins and it has fats and sugar, the same like milk, but not the same protein. And enzymes, when they work, they are in most of the cases very specific. In the case of chymosin, chymosin can cut casein, and it doesn't cut whatever, it cuts in a specific place, and that is why it has this effect over the milk, but it doesn't have any similar effect in almond milk. You can coagulate other proteins different than casein. Actually, many proteins will coagulate when you heat them, when you boil them. A lot of the things that we do when we are cooking is coagulating proteins that are in our food. And that is just the effect of the temperature. But casein and almond milk, actually, the proteins in almond milk actually are not that susceptible to coagulation by temperature. However, they are susceptible to coagulation by other process. If you feel like it, if you want, this is a totally home experiment. You can at home test the effect of the temperature in this experiment. You could test the effect of the pH by adding baking powder or lemon juice or vinegar to your experiment and see what happened. That I'm sure if you are interested in this kind of things may lead to interesting things. This is the kind of things that we do in those days in SCHMAHL Science, home experiments, because we are working in Zoom. But when we are able to go to the lab, there in the lab, of course, we can use this same kind of experiments, but we also can do a lot of things, other things that are a little bit more sophisticated. We can use spectrometry for measuring the activity of enzymes, chymosin, but other enzymes too. We can extract and enrich, purify proteins. Here in the picture, I'm showing you the extraction and purification of green fluorescent protein that happens to be very nice to see, but we can use the same kind of techniques for extracting and purifying different proteins. And well, we can extract other substance, DNAs and nucleic acids and other things that we do in the lab. We actually have a lot of fun while we do that kind of things. In those days, we are having that fun in Zoom sessions, but we also can do it in the lab when we're allowed to go to the lab. I think I am close to my time.
Yes.
So I want to thank you all. Thanks for inviting me. And if there is any questions or comments, I would like to take them.
Yes. So please be brief with your questions because we're running a little late and the next speaker is already here. So if you have any questions, I would ask it right now. Okay, it doesn't seem like anyone's asking. So thank you, Mrs. Ortiz.
Thank you for inviting me.